Many of you are aware I am scheduled to give a talk at the ICE meeting in a few weeks on March 4th. It'll be very different than this presentation. In that presentation at the ICE meeting, I'll actually be talking about some of our data on mercury levels in fish in the St. Louis River and what we're learning about in terms of spatial patterns and species-based patterns, habitat-based patterns.
I'd like to be a week from tomorrow. It's coming up fast.
Yep. So this is much more sort of theoretical, talking much like Len was talking about different places, different ideas, different studies about what we know about how mercury gets up in food webs. So what influences mercury in food webs? So in my brain we can break it down into three things, only one of which I'm going to talk about. You know, there's the mercury bioavailability. A lot of that Len just talked about. What's the concentration of methylmercury? That's the methylmercury. My notation there. What's its chemical form in the environment? What are the pH and reducing conditions? We talked a lot about aerobic versus anaerobic, having oxygen versus not conditions as a important piece of that. Of course, we talked a lot about the sulfur and carbon chemistry, basic pieces of the fuel and what drives sulfate-reducing bacteria. Chelation, we didn't talk a lot about it all. I'll mention that in a future slide, but that is the ability for metals to bind to organic materials in the environment and therefore become unavailable. And we talked a little bit about DOC quantity and quality. I'll talk maybe a little bit more about that. On the flip side, the bacteria have to be active, so you could have lots of methylmercury, but you could have lots of mercury available, but not much methylation if the bacteria aren't doing what the bacteria would want to do, which is to methylate. So depending on the temperature, the pH and redox conditions, the electron availability for the bacteria and the bacterial community itself are all drivers of the rate at which mercury can get up in the food web. And beyond that, there are a series of food web properties that actually drive what we measure in biota. They include ecosystem type, the inherent productivity of the ecosystem. Does it have a lot of algae or a little? The relative land cover, we talked a bit about that. I'll talk a little bit more. How much forest and wetlands are in the area. What are the food web pathways that are active in the ecosystem? What are the trophic level? In other words, where do they feed on the food web? I'll talk a bit about that more. The age of the organism, that's most prominent in fish because they're long-lived. And of course, with respect to fish, also the growth rate. And so I'm going to really focus for this talk on this part of the story. And if you have a question, just stop. If I use a word you don't know, if I use a plot you don't understand, we'll talk about it. I'm going to show you a lot of original data. And then one of the themes to be is also look at the kind of data we have to work with. It's often nice to talk about trends and patterns, but I think it's helpful to think about what we're extracting and the kind of information we get from the environment. So this is sort of a high-level picture of mercury in food webs. We've talked about zones where we have methylation and demethylation occurring, particularly in sediments where we have low oxygen. We talked about the major sources, elemental and divalent, or Hg2, coming from the atmosphere in the form of both wet and dry deposition. We talked about the role of wetlands in terms of methylation, so this figure should start to look interpretable to you at this point. That a lot of that methylmercury comes off the wetlands and out of the sediments. Some of it degrades, as we talked about, back into elemental mercury, becomes ultimately biounavailable. Some of the methylmercury gets uptaken by algae. That algae is then consumed or deposited and gets into organisms and low on the food web, and then they go up. And so I'm going to really be talking about much of this piece here. But hopefully now that figure is starting to look a lot more recognizable to you in terms of those processes. Notice too that along this pathway within the water column, there's one of them we talked a little bit about is the loading from the land, that the mercury can come in off these tributaries through either flushing events or regular discharge events. That mercury actually cycles within the water itself between different states, which affects so the relative chemistry can drive it more toward a divalent or an elemental form, and then that is changing with the sediment. So even if I take away this part of it, the wet dry deposition, we can still have mercury stuck in the water environments. And in some cases, that's an obvious impact, what Lynn was saying about reservoirs being storage for mercury. If I shut off an atmospheric source and I've created a system where there's legacy mercury involved, it may mean that becomes a ongoing and possibly active source of mercury to that system, even without some of those other inputs. So there's lags and storage systems built into these that add to the complexity. Before I want to talk a lot about this, I'm going to try to give you a primer on how to be a food web scientist, because I'm going to use some technical terms. So let's just kind of get all up to speed on some of the basic concepts we use in characterizing how aquatic food webs are put together. Effective trophic level. This actually goes back to the idea of a Minnesotan back in the 40s. The idea that we can actually break down food webs and organize them by trophic level. And this represents basically one step of energy transfer, is how you can think about that. So trophic level 1 are always primary producers. For most ecosystems on the planet, those are photosynthetic autotrophs. Plants that are fixing carbon using the energy from light and converting it into carbon if it's available to the rest of the food web. 2, these are your primary consumers. Think small invertebrates in aquatic ecosystems. Of course, some fish can eat algae. Mostly it's benthic invertebrates and small grazing invertebrates. And then it goes up from there, animals that eat those animals, that eat those animals, and so forth. Theoretically, the peak of most food webs is around a trophic level of 6 or 7. That's because with each step in this food web, the trophic transfer efficiency, the amount of energy they can move up a layer, is only about 10% on average. So you have to have a decline in the biomass. There's a limit to this based on basically thermodynamics. The idea of effective trophic height, though, and I think this is what's a little more foreign to people, is that you can have an animal that's here at a trophic height of 3.5. Intermediate. So this would be an animal that eats these bugs, maybe a small fish, and in the aquatic food web, this might be a bigger fish eating that smaller fish. But a fish here might be eating some small fish and some bugs. And we might estimate an effective trophic level of 3.4, for example. Make sense? So we actually can do better than just 1, 2, 3, 4, 5, 6. We actually have this idea of effective trophic level, which represents sort of the weighted composition of the diet. That's how we usually estimate it. So if it was 50% bugs and 50% fish, it'd be about 3.5. What complicates assigning of trophic levels and understanding aquatic ecosystems is that they actually integrate energy from a lot of different sources. So this is a classic food web paper, and I just wanted you to focus here on the bottom of this. This is from a couple of ponds, not particularly complex food webs, but even in this very simple food web, phytoplankton, bacteria, so this is algae in the water column, bacteria, paraphyte, that's algae that's on the bottom, and litterfall are all contributing in varying amounts, and the arrows represent the relative strength of that connection. So if my simple model of what gets into the food web is, it gets into the water, there's a phytoplankton on the water, the phytoplankton uptakes that contaminant, and then it gets in the food web, even that simple model is incorrect for most aquatic ecosystems. Even these simple ones, they're actually integrating energy from the terrestrial environment, from the open water, from the bottom of the system. So you have to understand where all those energy coming from and characterize them to understand that food web. We often teach as something much more simple, but this is much more typical. So if we think about a lake embedded into a landscape, we often think about the different rates of productivity of those. And so this is just an example of, this is relative productivity, high productivity, low productivity. So our forest and our herbaceous systems and our upland are relatively low productivity compared to these fringing systems. But what we have is we have some transport of that material coming off the land into the water. Some of that is bioavailable, some of it gets consumed. We have wetland material, which can actually be consumed, and that can be consumed a couple of different ways. The material can die off and be flushed out into the lake or the river, or it can flood and animals can actually move into that zone and feed on it directly or in and around it. So we have these highly productive wetland environments, and then we have this offshore pelagic environment, which tend to be relatively low productivity. So all these things are contributing. And they have different rates of productivity across that landscape.
So do the bars there represent like total productivity? Is that what those are?
Yeah, that's a metric tons per acre per year, sort of big global averages over lots of studies.
Wetzel, 1983.
So let's break that down even more, in case I hadn't visited you the array of autotrophs that can contribute to the food of an aquatic ecosystem. Food web ecologists actually break that down further. Besides our sort of planktonic sources of phytoplankton, we have algae that grow on rocks. We call them epilithic periphyton. We have algae that grow on plants, epiphytic periphyton. We have algae that grow in the sand, epipsamic periphyton. And we have epipelic periphyton, which grow over the sediment. So we actually break these down. Why do we break them down? Because they're actually they're very different chemical environments. You can tell that living at this interface on a rock here has a very different chemical environment and the relationship to this cycling of oxygen and carbon and nutrients than one that's hanging up at the top near the light. They're different light environments, probably different oxygen environments, different chemistry environments. So food web scientists are often interested in their different sources. Let's make it complex. Let's talk about the most complex kind of ecosystem we know of, and that's a coastal wetland. And one of the interesting things that's been going on in this discussion of the St. Louis River is we have to recognize that the estuary is a coastal wetland, which makes it one of the single most complex ecosystems on the face of the planet. We often break these into some standard categories, or just to understand them. We have our offshore. Usually we have this zone of emergent and submergent marsh that's generally out to about 2 meters deep. That's a pretty good generalization across the Great Lakes. After that, the bottom is light inhibited. We no longer get plants that are able to root on the bottom. There's just not enough light for those plants to live. So we have this deeper submergent zone of submergent marsh plants. Their life history is basically entirely submerged. They are wholly in the aqueous environment, up to an emergent marsh, which often includes some submergent species, but many of these plants are actually utilizing, they have structures that extend over the water surface, flowering structures, maybe getting some of their carbon from the air, rather than just the water or the sediments, up to a typical fringing marsh or meadow, wet meadow, where we have a high level of organic content, and then often there's, especially up here, we have the bordering cedar swamp. So we have multiple ecosystems side by side, different levels of productivity, different sources of energy. The St. Louis River is actually a drowned river mouth. They're actually a little different. This is probably not a great depiction exactly of the St. Louis River situation, but what's different on drowned river mouth is that because of isostatic rebound from the recession of the glaciers, the eastern end of Lake Superior is actually rising, rebounding slightly faster than the western side. And so in these systems, literally, it's a river that's backfilling these drowned river mouths. And the St. Louis River is an excellent example of that kind of system, which means they tend to be accreting systems, which is why this is sort of all this massive peat. We don't necessarily have lots of peat on St. Louis River, but it's an accreting system. It's growing. The wetland is actually, these systems are very effective at maintaining the wetland and capturing sediment, which means they tend to be, your wetlands tend to be organic rich.
There's lots of sawdust in there.
Thanks, Gary. Yeah, and humans have added to the accretion, of course. Sawdust, taconite pellets. If you go over to certain parts of the harbor, you can also find accretion of metal parts and machineries and brick. So we've done our share of building up the marsh surface around the system. So just to remind you, at least the St. Louis River, the lower estuary part, you have these large marsh features like Spirit Lake, Kankama Bay, and Louise Bay. And of course, they also have a very different character and a set of ecosystems in the open water. So you have to keep that heterogeneity in your mind, that the systems respond to all these different ecosystems, and they have different processes and different habitats different rates of storage of organic matter, different chemical cycling. And so if you thought of it as sort of a simple, this is a river, and then you missed one of the points of today's discussion, which is that it's part of what we're composing. This is some of our own work on the St. Louis River. Actually, a series of wetlands around here. This is the St. Louis River plot on the top. It's hard for you to read. These different boxes represent different algal sources to the food web. These little circles represent different species of fish. This is from the river. This is from the lower part of the St. Louis River estuary. This is out in Lake Superior. And the take home for you, not to understand all the details of this plot, is but to recognize that the relative contribution of those different algal sources vary by place in the river and by the species they're contributing to. So all these fish are not created equal. Some rely more on periphyton and benthic sources. Some rely more on water column sources. And that may change depending on where they're located in the system and the ecological features of that system. So these can be very spatially complex.
Don't the fish seek out these different places according to their species?
Yes and no. Some species have very narrow habitat tolerances and will continually adopt a very similar behavior independent of the environment. But most fish we have, because these rivers are variable environments, the fish have wide tolerances. And so many of them are adapting to local conditions and will differ their behavior based on what's available because they are tolerant. When you think about a system like a river where you go from high flow to low flow, under high flow it has much higher levels of turbidity, higher levels of sediment, much higher river flow itself. Under low flow the sediment drops, the water becomes more clear, the water becomes warmer. These fish have to be able to survive all of that. So most of the fish we have are quite adaptable. This is just a sort of a classic plot, but just to reinforce the idea, trophic efficiency is how much biomass gets transferred up. And so this is a plot. These are for a bunch of mesopelagic fish in the deep sea. Just as an example, this is the biomass of those fish in the deep sea compared to the overlying ocean production. It's nice because it's a very simple system. It's the open ocean. You don't have all these fringing sources and complexities. And you get a very nice straight fit indicator. There's a very constant efficiency up the trophic level. So we can measure that. We have ways to do it. And it's an important feature. We'll talk more about efficiencies later on. The last thing I want to mention is the idea of trophic status. So trophic status talks about the relative productivity of an ecosystem. It matters for methylation. This is the classic lake trophic state index. Most DNRs in the upper Midwest, PCAs, and those type of organizations use this. Essentially, the idea is if you know something about the transparency, a little bit about the pigment from algae, the chlorophyll, and something about what tends to be the limiting nutrient, total phosphorus, then you can correlate that to a trophic state. And so what you tend to find is that the lower the transparency, the higher the chlorophyll, in other words, the more algae, and the higher the phosphorus, all to go together for representing much more productive systems. And on the backside, systems that tend to have high transparency, low chlorophyll, and low phosphorus tend to be not very productive. Any given lake, you may find some discrepancy in there, but as a general working tool, the trophic state index does okay. But the simple idea is that we can then classify these systems as low productivity or oligotrophic, medium productivity or mesotrophic, high productivity or eutrophic, and then hypereutrophic is extremely high. And those can be natural or anthropogenic. There are naturally productive systems, and there are those that are only not productive because of human additions of nutrients to the system. So measuring mercury in food webs. To measure it, it takes a little more. Let me just stop. Any questions about all that? That was a lot in a really short time.
Could you share the studies that they came from with us?
Yes.
Thank you.
Yeah, I can write a bibliography. I can actually write reprints of almost everything.
Thank you.
It's just as PDFs. You sure? Before I move on, last chance.
Do you measure methyl mercury?
Do I? No. I have colleagues who do.
At the lab?
Right now, we're working, we use 2 labs working with MPCA, the Ag Lab, the state Ag Lab, and we're working with Gustavus Adolphus, Jeff Nice's lab. You'll be sad to know that the DMA, there is not a DMA right now at the lab. There's been a transition to a much more big focus on emerging contaminants. The interest now in mercury actually comes because out of Chicago, there's much more interest in needing scientific support to get the areas of concern cleaned up. They see a finish line in sight, and they've asked the scientists to come help push them to the finish line, which has sort of brought back an interest in legacy contaminants on the research side.
You may get to this later, but there must be a dramatic shift in the rate of processes going on between winter and summer. So you have kind of like 2 different systems between winter and
It
summer.
is, yeah, we'll talk about that too, yeah. We'll talk about temperature and seasonal differences, yeah. So one of the, and that's true in these food webs too, a lot of food web processes. In fact, this is a huge gap in our scientific understanding. When we look at these sort of dynamics, this is during a growing season. There's some legacy material from all of this that helps fuel the system through the winter. We have very little knowledge of what those processes are. Presumption is metabolism in most aquatic animals is fairly low because life is close to freezing. Water temperature is 4 degrees Celsius, or slightly above. So the presumption is that a lot of animals are running on reserves, but every time someone goes looking, they find that's not true. That under ice algae or some other feature is actually producing an interesting level of productivity in the winter.
How far does mixing with Lake Superior water go back up into the estuary, and how much of an effect is that?
Oh, that's one of my favorite questions.
Is it?
So we've measured it. There's 2 features there. One is the pumping action of the seiche. So the seiche is a wind-driven change in water level. We'll talk about changes in water levels later. On our system, on the St. Louis River, the periodicity is there's not a single period. There's multiple periods for the seiche. The one that's probably most well known occurs about every 2 hours. But that wall of water propagates up pretty much to the 23 bridge. If you go back to the old hydrologic study station, and if you're up there, you know this. If you've been there out in the summer, you've probably seen the river run backward up there. That said, remember, you have to think about the seiche as really the result of 2 balancing forces, water coming down and water being forced in. So if I can force the water backward, that means that that volume being forced in actually has to be larger to get back up in there than some of the water coming down. So in the summer, that's true. The water being forced in by wind is actually bigger than the volume coming out of that river, and so there's actually landward encroachment of lake water. So if I go out there and measure a conserved tracer of lake water, if I go out in July and then August, September, I'll actually find out that there's a greater influence further up the system. We've estimated that the average contribution of lake water at High Bridge or at Black Neck Bridge is about, on average, annually, 75% river water, 25% lake water. But it can be, we've measured it up to 80% lake water there under very low flow conditions, as measured at the gauge scale. So it can be a tremendous influence. Yeah. So it changes water level height throughout the system because of the wave that propagates up. The mixing is limited. We've never measured significant mixing much past Spirit Lake in terms of lake water. So you can see the level will change, but you won't see much evidence in terms of conservative tracers of lake chemistry influence.
So how does that affect this particular schematic here?
Right, so we thought about this a lot. What we've always been looking for is some evidence of lake being moved in. We don't see a lot of it actually in terms of energy. And if you think about it, that's probably not shocking in this system because the lake is so ultra oligotrophic. There's way more local production than there is in the lake. The concentration of food in the lake is relatively small compared to what's in the St. Louis River estuary. So even if I bring a lot of water in, it doesn't add that much food. That makes sense. So we've looked for it again and again. The signal's small. I mean, here's a little bit of it. Here's some evidence the rainbow smelt had some connection to that lake energy. But
One more question before I forget, you have to discuss it now, but the ships that transport iron ore or whatever bring about 15 billion gallons of water,
balanced water, yeah.
That's just a small amount, is that ever, have you ever seen an effect of that or anything like that?
So there's a group actually trying to measure that. No, you really can't. It's really, I mean, if you were there right under the spout, you could see there's a small difference, but the dilution is rapid. It is a drop in the bucket.
What about the
For
different?
the individual pumps running when they do. Cumulative may be bigger, but when a pump is running, it's actually not that much more river water.
What about the creatures, maybe different, fish, and as you go down the food chain, you bring new deposits in, well, some of that.
Right.
Does that affect your analyses at all? Just not necessarily the amount of water, but the amount of mussels or
from the discharge?
Yeah.
No, not really, but again, if you remember, they're being asked, a lot of the exchange is occurring in relatively low productivity systems compared with this. But it'd be a very hard effective test. We're actually trying to do it with invasive, just trying to see if we can find invasive near there. And these are very small scale ecological gradients, extremely small. It'd be very difficult to detect against sort of clear signal. Anything else? This is a really important plot. Kind of what Gary's alluding to. So this is the level of mercury we tend to find in water, total mercury, and the portion that's methyl. Notice they're often order of magnitude or more difference. Extremely low concentrations. So this is a log plot. So each one of these is an order of magnitude jump. So you can see that where's the biggest gap we typically see in aquatic ecosystems is right here. Very low methyl in water and much, much higher. Orders of magnitude, 5 to 6 orders of magnitude higher sometimes in the algae. So this is where the action really is occurring. Is this first step. That's the massive sequestration of methylmercury into the environment. It then moves up into the food chain. But you're still not done. So this is zooplankton, our summer primary consumers, small planktivorous fish, piscivorous fish, and then these are loons right here. And of course they're eating the fish. But notice even then, the spread of those boxes are themselves an order of magnitude change. That's tremendous. When we're talking about state standards at about between around 0.2 versus 0.1 ppm is safe to eat. And we're talking about typically clogging and variation, it's an order of magnitude. Which means that here, a small change in the accumulation factor between methylmercury in the water and phytoplankton has tremendous implications for the whole food web. But it also means that very small changes in this part of the food web can be the difference between meeting your state standard and having fish that's safe to eat and not meeting it and having fish that aren't. So these are, anytime we start talking about things that work on these sort of big log scales, the science gets really tough because there's so much inherent variability that dealing with very small targets that could be 0.2 and 0.3 ppm against this level of process is very difficult. Very difficult. The other thing I want to point out about this plot that's very typical, here's the percent methylmercury. And of course, in phytoplankton and of course in water, water is mostly, a large portion of that will be inorganic. But it is that methylmercury that actually bioaccumulates. If the animal consumes inorganic mercury, it has ways to get rid of it. Over the gills, we can excrete that. It's the methylmercury that binds up the tissues and hangs out. And that's why it bioaccumulates over time. And so you see that methyl proportion going up much higher relative to the total mercury. So we usually measure, because it's a log process, this is sort of the horrible way that you see this data presented all the time, is this log VAF. Well, what's a VAF? VAF is the amount of methylmercury in a tissue that you measure relative to the amount of methylmercury in the water. Now notice on the log scale where it's falling. Hundreds of thousands to millions scale. Log, that's 5 and a 6. That's 5 zeros, 6 zeros. Does that make sense? So we're talking about accumulation factors over water that are in the 100,000s to a million times more concentrated than water. Tremendous accumulation factors. This shows some variation among two different tributaries. This is a study from out east, from Maryland area, between just some typical stream organisms, some caddisflies, some stoneflies, crayfish, a little dace, some minnow, and some different size trout. And what you should automatically pick up is that the bioaccumulation factor among our lower trophic level animals varies quite a bit. Nearly an order of magnitude, and it varies by system. And those are just two small streams measured. So the implication for these fish then, and what's safe to eat, is tremendous. Whether you eat this or whether you eat that, makes a big difference in how much mercury that fish is going to have in it. It also matters how much methyl is in it. And now I just told you that mostly it's methyl. In consumers, that's true, but even that's variable. So this is from the same study. They actually took the time to measure both the proportion that was total and the methyl proportion. So these are some different measures from some different streams for crayfish and sculpin, dace, trout, again. And this is the same. Now these are different locations along the stream. Now that's in percent. And you'll see that even the percent varies across sites. So some have more methyl, some have less. Some have 50%, some have about 75%. And it can be dramatic. This sculpin, one sculpin had 25% methyl, another 90%. So if a big fish ate that sculpin, this one is sort of less problematic because so much more of the mercury in it is inorganic. I say it's not a problem, but the inorganic mercury, it has a way to shed. That methyl will bioaccumulate. So it doesn't just even matter which thing you eat, it could matter the conditions and the timing of what you eat and exactly how much, depending on the time of year and the metabolism of the animal and what it's doing, how much methyl versus total mercury is in it. So these things aren't necessarily straightforward. This is a similar plot shown here for the second stream. Paraphyton, bryophytes, so cranefly, caddisfly, mayfly, stonefly. And again, you can see some big variations. Here are two different dragonflies. In both the methyl mercury content and the relative amount. But those are the two typical ways we measure it. So you almost always, when you look at food web studies, you'll either see a VAF produced or some concentration. Now, a lot of studies, and this is just out of convenience, it's really expensive to measure methyl mercury. And ecologists would like to have more money. That's my pitch for the day. We usually don't. So it's often convenient just to measure the total. It's much cheaper, it's quicker. It's analytically robust. And then we sort of presume then that by and large, the total mercury is a good measure of the methyl mercury. We're using it as a proxy, basically. And you can see that sometimes that's a pretty good assumption, but sometimes it isn't. And so when you're reading the primary literature trying to understand, you should know which form they measured and how they're working. I know on the St. Louis River there's a lot more interest now in being much more careful. Long-term monitoring kind of has to be dirty and cheap in some sense. And so much of the long-term monitoring is based on totals, not methyl. So for a lot of the long-term monitoring we have that the state does, both Minnesota and Wisconsin, this is true for EPA too. We do the same thing for our Great Lakes monitoring program. We don't often measure methyl. We tend to just measure total and presume that it's a good proxy. So here's the meat of it. Everything that can influence mercury in aquatic food webs. water chemistry, trophic status, land cover, water level fluctuations, that's sort of the reservoir discussion, but we'll kind of push past that too. Aquatic ecosystem type, and then visual deposition pattern. So we're going to just kind of walk through those one by one. Again, before I dive into this topic, let me ask any questions about measuring mercury, accumulation factors? Water chemistry. We talked a lot about this. I'm going to kind of blow through this. We've been through this. I gave you a diagram at the beginning that looked a lot like this. This one gets into more specifics. But one thing we haven't talked about, I'm going to get more into DOC, so I'm going to point this out. So here's sulfate-reducing bacteria converting divalent mercury into methyl mercury. It shows it here both in your pond or lake sediment and up in your water column. But an important piece of this cycle that we're going to talk a little more is this idea that mercury readily binds to particles, organic particles. And it's been a huge part of the issues with understanding the mercury environment have revolved around that relationship. I'm not an expert in that. Actually, the great person in the region that talked about that is Dave Grabenhoff at the USGS. Dave is actually a partner in some of our St. Louis River projects and is a world-renowned expert in some of those processes. That's kind of one of his passions, is understanding, teasing out those relationships.
How do you spell his name?
Grabenhoff, K-R-A-B-B-E-N-H-O-F-T. He's at the USGS down in Madison.
Thanks.
But he is a partner on some of the St. Louis River work we're doing now. So we're getting Dave more and more involved up here. This is really a classic mercury paper. So I'm going to point out a couple, we'll take a little time on this slide. There are a couple, so there's that term again, BAF. Now you know what it means. See the logs, we're in that same order. There's thousands to millions to even 10 million plus accumulation. So this is a study of, this is actually a big summary of a bunch of data sets from northern Wisconsin. And a couple big patterns emerge. One is that the fish mercury, total mercury, declined with increasing pH, or if you want to think about it, was augmented as the lake at decreasing pH. The more acidic the lake, the higher the mercury in the fish. DOC, the higher the DOC, the more the mercury in the fish, and it increases almost fourfold. More like, probably like 2.5-fold on average across that data set. And this is the interesting thing. So I just told you that mercury goes up with DOC. But I'm telling you something confusing. I'm going to tell you that BAF actually goes down with DOC. Why? How is it that systems with more DOC can have more, the fish can have more mercury, but it's actually that the BAF can go down? Well, this is, if you think about this, this is really about efficiency. How efficient is that mercury getting up into the system? The fact that the BAF is declining tells you that these are less, that with this DOC, high DOC system, are actually less efficient at bringing that mercury up into the food web. That's because the DOC actually interferes with it. It's a great carrier, it's a delivery system for the mercury. The mercury is actually bonding with the DOC, but at the same time, you have to break it out of those bonds. And so we see efficiency dropping. So this is a very typical finding that you see that the efficiency for it. Now you can see that the effect is still net positive. In other words, despite the decreased efficiency, mercury is still going up. So that's a common feature we see in these systems. I also want to point out to you, just again, come back to the sort of the real data check. I mean, these are lovely lines, but notice there's a lot of scatter around. Very typical mercury data sets. I'll show you some of that. So we can pick out big signals, but they're big signals. Even within a pH of 6, you can see that there's a twofold variation in the amount of mercury in those lakes. So it's a big signal of a big area. But if I had to precisely predict for you the mercury based on any one of these factors, chances are I'd be dramatically incorrect. At heart of this is this sort of problem. I think if you think about the underlying chemistry, I would recommend you think about it as competition. That the mercury molecule is so reactive, especially in that divalent form, that it's constantly in competition between a particle phase, a colloid phase, bonding with various organic species, and then also attaching itself to inorganic species. So it's constant competition. So the local chemistry environment has a big input, which is why we can see with a simple chemistry parameter like DOC or pH, ecosystem level effects. It's because of this competition. You can't do all those things. It goes one way or the other. It's a competition. Another example of the same thing though, if we look at the carbon, the DOC against filtered mercury just in the water, we get positive relationships. Depending on where we are, Vermont, Colorado, Puerto Rico, Wisconsin, we can get different slopes, but we see the same pattern. If we look at it in terms of the particulates and the unfiltered, same thing. It tends to be a very positive relationship. Nonetheless, here's that BAF again. This is from zooplankton. This is for seawater, but declining as a function of that DOC. Also, point something else, just because we talked about mixing the St. Louis River. For those who don't know, Lake Superior is a very low DOC system. Obviously, it's quite clear. Superior Bay is right here. It's about 3 milligrams per liter routinely when we measure it. Under spring discharge, the upper river is out here. There's a huge DOC gradient within the St. Louis River, tremendous, because of the mixing of low DOC Lake Superior waters with the St. Louis River. This is in the estuary. This is the upper watershed. Most of the rest of the watershed, you tend to clock 20 to, you get 40, you get spot samples, 100 milligrams per liter DOC out there. But she telling you, St. Louis River tends to run between 20 and 40. It's kind of a good ballpark. But in the estuary itself, we can actually get tremendous drops. So we have these microchemistry environments that also become an issue to think about. Trophic status of the water body you're in. Another kind of chemistry paper. This is measuring mercury in fish. This is parts per million. So this is 1 ppm is of course not good. But what you see here is that this big wedge-shaped triangle plot is sort of a classic limiting factors plot. We see the kind of plots in environmental science all the time. Well, the story here, though, is as the total phosphorus in water, in other words, as the lake productivity increases, that becomes a limiting control feature on the methylene fish. So if I have a really productive lake, I'm not going to have much methylmercury in the fish. Why is that? It's not well understood. The reigning hypothesis is called the dilution hypothesis. In other words, we have a lot more cells in the environment, and so those cells are basically among them diluting the amount of methylmercury. Because they're highly productive. And uptaking lots of carbons, there's less mercury per carbon unit, if you will. And so that dilution extends up the food chain. That is a hypothesis, so it's well understood at all. But it's a common feature we see in environmental data sets.
Does that look like Lake Tanganyika up there by Midtown? They have that
Oh, yeah.
highly productive green lake.
I know, yeah. So, it's, I don't know, I have never seen chlorophyll for that.
It just looks like it.
Yeah, yeah. A couple other things that this paper looked at. This is a common health metric, about 0.3 ppm. In Minnesota, we tend to keep it a little lower. But so for fish less than 0.3 ppm, fish greater than 0.3 ppm, so this is DOC, so higher mercury, higher DOC, higher mercury, lower pH, and then after acid neutralizing capacity, basically the buffering capacity of the system, that gets into sort of the electron potential. And in this case, lower capacity, higher. So these are all sort of all flow out of discussion Len was leading about. This idea that there's some basic chemistry are big drivers. Again, though, notice that even in these plots, there's overlap in the environmental conditions for the same fish. So I can have a pH of 6.5, and then I get some of the fish in the low and some of the fish in the high. These aren't necessarily clean or easy. Land cover. Lynn talked about a lot about this. I won't talk about it much. I like the schematic quite a bit. So he talked about how conifers are actually better than deciduous trees for getting methylation. So you'll see that there are, the stippling means it could be methylating environment. So you see the 2 versus the 1. It's there, subtle, but it's there. The wetland highly stippled, there's our big spot. The stream less so stippled. And then in the lake, you see that a lot of fine stippling here in the sediments. That's usually, that interface is usually our big methylation layer, but even then they recognize that some potential is available in the water column, depending on stratification.
I needed that slide.
Yeah. So, yep. So very, very. So there's sort of our ecological frameworks that we're often working off of. So the theme of this talk of this morning is so far is it's always more complex. So it's always more complex. Another way to think about it. Here's our forest. Here's the augmentation of wet and dry deposition through litter fall. We have increased rates of mercury deposition in around our forests. We have our methylating hotspots and our frigid wetlands. And Lynn talked a lot about flushing. This is a really important feature, actually. The local hydrology is a huge driver. You can go to wetlands. There are a wealth of studies showing that you can have a wetland that is a perfectly good methylator that has very little contribution to a local water body. And the reason why is you need water to go through that wetland and transport that methyl mercury out into the environment. So you need a flushing activity. You need enough groundwater flow or subsoil flow through the groundwater here to get that stuff out. Which means that shallow water flows have a much bigger impact than deep flow paths. Deep flow paths will contribute very little. Shallow flow paths will contribute much more. And then the volume of that path makes a big difference in how that wetland will interact with the permeable water. Those are really hard studies to do. Groundwater studies of water flows through wetlands are really tough. And if you're trying to get at these flows, you need wells dug for decades because these are looking on decadal time scales. So these are really tough studies to do. Lots of time, lots of energy. In this, Evers et al. Was proposing kind of their own version of sort of what's a vulnerable system. They flagged sensitivity less than 30 micrograms a liter total phosphorus, DOC greater than 4 milligrams per liter, pH less than 6, and acid neutralizing capacity less than 1,000 is their sort of way of thinking about what's a vulnerable system. For locally, to put that on your brain, thanks to efforts to reduce phosphorus, both from the landscape and from human sources in the watershed, the St. Louis River is now routinely below 30 micrograms per liter TP. So the nutrient load has declined dramatically in the system. We're approaching a fairly healthy, but that makes us more vulnerable to mercury accumulation. DOC is routinely above 4. pH is actually high in our system, 7.5 to 8.
Really?
We're a high DOC, high pH system. Water level fluctuations. This is a really complex graph that describes exactly the story Lynn was talking about with reservoirs. This is a well-known phenomenon, very well studied. If you have a reservoir, and you have atmospheric deposition, wet and dry, and then it gets drawn down for a period, these sediments, oxygen returns to them. And if there were previously no oxygen, there was sulfide. Now there's now new oxygen, there is sulfate. That sulfate, when they refill the reservoir, can be an important feeder for methylation. So you get a huge amount of methylation on those inundated sediments. And that means that shortly afterwards, in that first year, you get a massive spike in mercury in the fish, and then declining after. Well-known phenomenon related to reservoir management. So there's lots of questions, though, about the periodicity of this. So this is true. I mean, this is an observed loss. But does this have to happen for 5 years or 3 years, seasonally? That's much less known. Generally speaking, folks have looked at seasonal changes in water level on lakes, and also saltwater estuaries, have found that the effect doesn't seem to be all that dramatic. This is a study done out of Maryland. They're trying to compare some of their dam systems to some of their estuary systems. So here's their reservoirs, very big slopes, meaning that there was, this is for a size-fish relationship. So there's a rapid increase in mercury with size. These fish basically have lots of mercury. And when they compare that to fish in the tidal fresh waters of the Chesapeake Bay, their overall mercury was much lower. Now there aren't great controlled studies on this. There are not a lot of dam operators who sort of willingly altered their dam regime and looked at methylation. Of course, because of this problem, it would be a really hard study design because you'd have to deal with the lags to understand the true effect.
Quick question.
Yeah.
Are the fish populations changing here, or is the mercury changing within the fish population?
That's a great question. So the other issue with doing a study like that is if you're going to say, I'm going to pick 2 different reservoirs or 2 different types of systems that have different water level fluctuations, I'll pick the same fish, largemouth bass in that case. But they're embedded in whole different food webs and different processes. So now there's an apples and oranges problem with the design. I can't exactly isolate the effect of the water level change against the other changes with the ecosystem. But in fact, changing the water level changes the ecosystem. So it's a really tough problem. By and large, sort of the consensus is that short-term, seasonal, or daily changes in water level are likely to have much less effect than the big cycles you see in most reservoirs, which are annual or longer between fills. But that doesn't mean there couldn't be a condition in which that was not true, where a seasonal cycle could drive some enhanced methylation.
What's this one from? This
Oh,
demo?
I'm sorry. Yeah, they just cite. For those
It's a good one.
who want a really good read, I will highly recommend Mercury in the Environment. This is a recent book, came out in 2012. Michael S. Bank was the editor. You can find it on Amazon. I think it
Sure.
costs. No one wants these things used, so I think I got it for $25 or less.
Mercury in the
Mercury in the
Environment.
Environment.
By who?
Michael S. Bank is the editor. It's a series of papers from contributing authors.
Thank you.
This one came out of there.
There it is.
What's the
Okay.
last one?
Bank.
B-A-N-K.
Bank.
Bank.
I'm trying to make a list of
Sure.
things that come up in here, and so, and send it out to folks.
So this has come a lot with respect to the St. Louis River. Folks wondering, does the SAISH enhance methylation? And I guess the simple answer is no one's looked at it directly. And they're unusual. And in fact, there's very few studies, and I have no finding studies in the Great Lakes looking at how SAISH patterns in coastal wetlands might affect methylation. But I know that comes up a lot, is whether or not the fact that you have routine periodic changes. And of course, those occur in different scales. We have the daily SAISH effect, which is hourly. But then we have seasonal, because of changes in lake level. And then, of course, decadal, that correspond to the rise and fall of the Great Lakes. So, all in all, what you tend to find across studies is that folks who try to compare aquatic ecosystem types, they tend to find routine differences. So this is for a bunch of different species. That's white sucker, largemouth bass, brook trout, smallmouth bass, northern pike walleye, lake trout, white perch. I should remember all those codes off the top of my head. They're down here. But by and large, what you see is that the reservoirs tend to run higher. And rivers and lakes tend to be a little more similar. More similar to each other than they are to the rest of them. So deposition matters. I think, step back, I sort of made this plot too small, I'm sorry about that. This is a very thumbnail depiction of the global mercury cycle. And what you should recognize is what human beings are doing globally are vastly perturbing it, and that's what the center represents. The industrial extraction of mercury from our deep reservoirs are, in other words, these are geologic scale reservoirs. Through burning of fossil fuel, through mining, releasing it into the atmosphere, and to the land. And these terms are as big as many of our global exchanges. So this is a huge problem. It has a local impact. So this is from the same one. They actually used a global climate model to sort of look at the different contributions. This is the global anthropogenic contribution. This is percent over natural. And so these are all hotspots for where humans are increasing it. Here's the Great Lakes. Here's the infamous Great Lakes to Northeast Corridor, which in every study, you look at Summarize the Nation, always comes up as our big problem area. Stretching from the Mid-Atlantic up toward Maine and then back toward Minnesota. And you can see there are also some major hotspots across the planet.
What's going on up there in the Arctic?
Yeah, is there something up in the Arctic? North America, Arctic?
I think that's just landmass. I think you're just seeing
Oh,
landmass there.
There's something in the Russian Arctic, or there's a paper out that shows that the flushing of the two main rivers in Siberia, Ob and the two others, are what's really causing the mercury to be high in the Arctic, in the Siberia
It's
Arctic,
interesting. Yeah,
as opposed to the deposition from China
right.
and places like that. They prove that.
And there's lots of concerns how climate change is going to inflect methylation through exposed sources, rerouting carbon cycles, means of reduction of permafrost, changes in hydrologic cycles through enhanced discharge, especially seasonally. What we're seeing is higher highs and lower lows. Lots of concerns that the climate change signals of methylation globally in mercury in animals. And then this one even parses out by contribution. So notice that it's interesting, European and Asian have huge far-field effects. The North American is actually much less of a far-field effect, but where it is an effect is mostly concentrated in our part of the continent. So this all adds up to the fact that we remain an area that also receives a lot of atmospheric mercury. Some of it's ours, for sure. A lot of it is
That's
far-field
a very
sources.
interesting slide when I look at it, because, you know, when you look at the Asian versus the North American of what we're receiving, where we are, it looks to me from that we are receiving just about as much from North America as we are from Asia. That's what it looks like. I don't know,
I have to go
you know,
back and look at the
right,
state numbers that I've seen.
Exactly, but, you know, when you hear people blaming it all on China, you know,
Well, so then,
it's like.
and I was going to go look it up, because you had mentioned a 90% number, which wasn't my understanding. The numbers I've seen for the state are actually about 30%
Not the
global, state-based.
But this is atmospheric. He was saying 90% atmospheric.
Okay.
Weren't you?
Yeah,
Yeah.
the
All
state
sources,
TMDL was
atmosphere.
based on the premise, and Ed Swain backed this up under challenge, that our deposition, 90% was coming from without,
Oh, outside
Our
of
border.
Minnesota.
but
Right.
it could come from the Ohio Valley.
Right.
Right, yeah, this is just from all North America. It could be coming from Canada for that
It is.
matter.
Well, Fairmont's
You know.
coming out of North Dakota and
Yeah,
Canada too.
yeah.
Yeah. And we have,
But
we are an
that's
area of the
different
country
than
that is,
China, you
at
know.
this point, we are among the most reliant on fossil fuel use and burning of coal for electricity.
Mhm.
In northeastern Minnesota, or in our region?
Just in the region.
So
We're very fossil fuel dependent relative
this
to other
this
parts of the country.
northeast region of the
Yeah.
United States, we're intensely
Yeah.
fossil fuel
So not an
dependent.
insignificant source for us is fossil fuel demand up here. Now, the mines are also, taconite production is also a substantial and measurable source of mercury in the environment.
Does the Enbridge refinery in Superior impact us?
Yes, so refining, in general, yeah, I don't have numbers for the facility, but the process refining can be a mercury source. Yep.
Significantly, I would guess, or not?
You'd have to weigh it against all these numbers. That I don't know. We have to figure out. There are probably some academic equations you could use given the nature of the facility, the processing method, and the volume, what their contribution is. They may be permitted for it. So they have to monitor it. So we should probably go to the NPDES system and look it up, actually. I've never done that. It could be done.
So if you put this on a time scale, if you're looking at what's happening in the Arctic by Siberia now due to flushing of the old mine sites, if you fast forward just 50 years, when all mining would have been done, had it been permitted in the Iron Range, we would be experiencing the same thing next period where flooding and stuff like that would flush out all these old mine sites and wash the mercury down into the lake or the estuary or whatever.
That's become a huge issue, is long-term management of those.
Well, there is no management, you've got billions of tons of sulfur. I mean, I'm just saying.
I think it's true. I think, and you'll notice this too now in the permitting. There's much more thought now about what to do with the waste rock in the mine afterwards, in terms of. So I think even like the one proposed for Wisconsin, some of the initial ideas were to tunnel or backfill as they went, so they wouldn't have these exposures and creating these systems like that. So, yeah. But yeah, if you left a big strong landscape like that, it has certainly potential for problem.
Well, there's 4.5 billion tons of that stuff up there, I'm just thinking that's not a small figure.
Yeah. This is wet deposition. This is total. I'll go either way. This is total. So this is total mercury. I just wanted to point out that the St. Louis River watershed comes out nicely as a purple relative to an area of green on our models. This is a smaller scale model than the one I just showed you. It's now a little dated. I don't think I've updated it recently. But purple is high and red is very high. And you can see that Minnesota's northeast corridor come out nicely, that we just saw on the other slide. But you can even see some regional patterns in Minnesota come up on this model. So recognizing the St. Louis River has some locally high emission. And then a more updated version of just wet deposition. These would be hot spots, but you can see again already an elevation to a mid-level range throughout this part of the region with less deposition off to the north, to the northeast. So, just a brief comment. It's really, some of these big patterns are hard to get hold of. One of the things that Len mentioned, it's really important to remember, is that it's not uncommon for these studies to measure mercury in the environment and total mercury, in methyl mercury, and find no relationship within a watershed or on a stream basin. Very typical. It's also typical to find not much relationship between total mercury and what's in the fish, for all the reasons we're talking about, all those variables that influence it. So these things are hard to glob onto, but it is interesting. At least at a regional scale, these patterns seem to matter. So these are state databases combined across a big swath of land, and carefully removing some outliers. There does seem to be a loose but apparent relationship that if you do have more wet deposition, the fish will have more mercury in them. So they're not utterly disconnected. They may be loosely and hard to get at, but over big scales, continental state scales, more deposition can translate to more mercury in fish.
Well, that's basically what our state mercury TMDL is based on.
Right. And this database is based on basically the entire eastern U.S. And you can say the relationship, including outliers, isn't good at all. And if you take us in the outliers, it's kind of okay. So it's a tough proposition, frankly. Yeah, you can see that the range we're trying to move, probably a couple milligrams, a couple ppm, 0.2, maybe 0.2 ppm, something on this range. And you can see that there's, within one deposition, you can account for all of that, one deposition regime. So, stuff. So what I want to wrap up on here is things that drive you really crazy. So it's not just that the ecosystem responds, individual organisms respond. So it actually matters the organism you pick up when you take these measures. Lots of things can just drive that measurement. So for example, which thing I measure? Do I measure the muscle, or the liver, or the kidney? One of the things you might see in this, for example, is that the liver and kidney tend to have fairly high inorganic mercury. That's because those are the organs that are being used to get rid of the mercury. So that's where it is. Methylmercury, very high in the intestine, moderately high in the muscle. That's probably the most typically measured thing in fish. But similar or less than, depending on which you do. And you can imagine a lot of studies actually use whole fish and some sort of version of all of those features. So just what did you actually measure from the fish? Makes a difference. Growth rate makes a big difference. This is really hard to read. That's the mean age from 1 to 8. This is a predicted mercury concentration based on a model. Every one of these data points represents a fish of the same size, 350 millimeters. Some of those fish are only a year old. Some of those fish are 8. So these are very fast-growing fish. These are very slow-growing fish. And the very slow-growing fish have much higher mercury. So if a fish is in a poor environment, besides the fact that the low productive environments tend to methylate mercury better, fish tend to grow slower in those environments because there's less food. And slow growth tends to promote mercury body accumulation in fish. So I could measure two systems that are nearly identical, except for one had slow-growing fish and one had fast-growing fish. That could be from a fishery harvest policy. And find out that in one lake, they had much higher mercury. Not for anything to do with the environment or the pollution. It's something to do with the harvest policy in the lake, driving growth rates in the fish. So growth rate matters, big time. It's a really hard to measure growth rate, by the way, super expensive, so we don't often do it. It takes a lot of tech time, and we have to kill all the fish to get a growth rate, a good growth rate, because we use the ear bone. We can do proxies with others, but they're not nearly as good.
Was that, Catherine told us that you did, that's what you did your, something about your thesis was something about.
I love fish odorless. Yeah, I do lots of things with fish odorless. Well, unfortunately, I have to sacrifice the fish. So, yeah. So often we use size and age as a proxy. They work pretty well. These are the relative amount of variation explained in mercury as a function of length. So in these burbot, 81% of the variability of the mercury was explained simply by the size of the fish. Some of these didn't work too well. So it generally works, but not always. Three spines to go back. Length and mercury were not related at all. And sometimes we use weight, and sometimes we use length, and sometimes length is better than weight, and sometimes weight is slightly better than length. We do this because if I pick up a sample of fish out of this river here, and I don't get a 6-incher, and I go down the watershed and pick up a fish that's a foot, I will get a different measurement. Because the fish is a different size. And as they grow, they accumulate mercury. So I have to control for that on the length. And it's very hard to find the same fish, the same species, the same size everywhere. It never works. We try, it never works. So we develop these relationships to try and put everything back together. But it's messy, and it doesn't always work. It's really tough. Also, trophic level matters. We talked about effective trophic level. The higher you are on the trophic level, the more you accumulate mercury. However, individual fish don't always feed the same. So this is just a plot, food chain length. These are perch. That's one variation food chain step. And you can see the perch generally fall in the middle. Most of them do. But some fed way low on the food web for being a perch, and some fed much higher. And walleye did the same thing. Not uncommon in fish. You pick up one individual in one lake, it's at one trophic level, it's at a different in another lake. Which means that I go to the same two lakes with the same pollutants and the same contaminants, and the food web have slightly different properties. The fish are feeding higher on the trophic level at one, they'll have higher mercury. Nothing to do with the environmental condition. Everything to do with the food web. Quite plausible. Like, numerous studies have found this as a problem, actually. So we try to control for that, but as it turns out, also controlling for trophic level is really tough. Because most of the time, if I pick out a 12-year-old fish out of the lake, I have no idea what it's eating for the vast majority of its life. I can develop proxies for it. We're getting better at that, but it's still really tough. Reconstructing the environmental history of that animal remains an elusive idea. That said, over big data sets, this is the entire western U.S., a bunch of states, fish data. I think these are all in mass. No, these are multiple species. They group non-piscivores and piscivores. So lower on the food web, higher on the food web. These are the ones that are high on the food web. These are the ones that are low. And sure enough, if you fit big curves over big data sets, on average, yes, the fish lower on the food web end up lower mercury than those higher. But gosh darn it, look at that scatter. But of course, this is controlled against length. So for the same size, if I pick up two 500-millimeter fish, on average, the ones lower on the food web would be lower in mercury than those higher on the food web. But you can find plenty of small dots up here and some big dots down there. And then, of course, it's not just where you feed on the terms of the trophic transfer, it's what you actually eat. So this plot shows the growth efficiency, and I'll add a site to this. But also, it also matters, besides the growth efficiency and the trophic transfer, the trophic level you're at, what's in your diet matters. So what are the things you are eating? Are they high, relatively high in methylmercury, or relatively low? So for example, go back into this reference. In this case, if I was feeding on these caddisflies in this river, I'd be much better off than eating those dragonflies if I was a trout. Much lower mercury. Feeding on the same stream. The change in diet can dramatically reduce the methyl content of that fish. And in lakes, habitat versus can matter a lot too. The carbon isotope is a marker of habitat use. These are more benthic. These are more pelagic. This is mercury. So you can see across species, the ones that are more benthic have lower mercury, and those are pelagic. But if a trout that moves between habitats, where it is. So you get a lot of variability in the trout just because of where it's spending its time. A trout that spends more time feeding in the shallow waters or on the bottom has lower mercury than one feeding out in the open waters. So within a species, within an ecosystem, I get variability based on the habitat use and behavior of the animal. So to wrap up. Going back to the beginning, the bioaccumulation factor is much greater for algae in food webs than for secondary consumers. So small changes in that algal EAF will have much greater implications for the food web than variation in consumers as a first order effect. That said, if we look at mercury concentration in biota, they're influenced by ecosystem scale, individual scale, and even tissue scale factors. Among those for ecosystem scale, the ones that are almost universally invoked: land use, wetlands and forests, level of mercury deposition, the kind of aquatic ecosystem, what's the structure of the food web, what's the local water chemistry. For individuals, we're always trying to control for the trophic level, the diet, and the age or size. To get good comparability. It's really tough to do those things. And it's extremely tough to do all of those at the same time. Rarely ever done. Maybe never done. It's quite a challenge. But the upshot is this: if you have multiple factors, all of these will simultaneously working on the measurement you take, what's in that fish that someone wants to eat. And these factors are operating on their multiple scales, and they can have complex interactions. One feature of the environment may enhance methylation, another may decrease it at the same time. which means that using pulling wild fish out of real ecosystems and interpreting the mercury concentration in them, it's quite difficult for characterization and for interpretation. It's a real challenge. So we keep all this in our mind as we go through this and do our best to control for these big factors. But it's no small task. Questions about that?
So based on your studies that you're doing now, ongoing studies of mercury having to do with nutrition of fish and then we who eat them, I don't know how recent most of the studies are, but we have this fish consumption advisory for mothers and children and pregnant women, and they are dated maybe 10 years ago. I don't know, but do these studies that you've been working on, do you recommend these? Is that how the state gets some of their
That's a good question.
guidelines or?
No, the state actually, there's a whole different field. This is a truly multidisciplinary problem. There's a whole separate field of public health specialists whose their job is to research how varying effects of mercury exposure impact people. And it's those experts that were appealed to, gosh, over 10 years ago now, to come up with what's a sort of a Great Lakes-wide common mercury standard, ecotoxicological standard. And that's what Minnesota adopted that. You can find documents relating to this on the Minnesota Department of Health website. So actually, they very do a nice job documenting it. Minnesota is among the most conservative approaches to that standard. And we're also probably one of the most aggressive for mercury monitoring. But my understanding, at least, is right now, is there's not been in recent years, maybe Linda was different, but I have not heard the public health side clamoring for a more restrictive standard. That hasn't been the debate. Most of the debate's been how to deal with this really tough pollutant and get it out of our watersheds and our fish.
So they're not really using the EPA studies that much, or?
Well, not for the health standard.
Yeah.
Not for the health standard. Now for understanding what's going on, they appeal to, the EPA works a lot with partners. So, for example, the EPA maintains the Great Lakes-wide mercury monitoring network for fish. That's housed out of Region 5 in Chicago. Beth Murphy handles that data set. But a lot of those fish are collected by state partners and submitted to EPA. Wisconsin and Minnesota have their own mercury monitoring programs on St. Louis River. They're not annual. They do what they can, I think is a nice way to say it. They don't have a lot of resources for it, and the resources are spread over a lot of water bodies. So they're responsive to those data, and it's upon those data that the current restrictions are enforced. And I would say from the data I've seen, they're appropriate. Most of them, certainly the walleye you wouldn't want to eat out in that river. Most people should have a restricted consumption. They're not doing that by and large, but they are. Limit your intake of fish.
So, a grown woman, 50 years old, what should she eat in terms of walleye from the St. Louis River that are, let's see, I'd probably catch one that big at most. That's my usual.
Probably between the once a month and once a week.
Of how much?
restriction Of a fillet. I think, and that is I believe it's, correct me if I'm wrong, I believe it's a 6-ounce standard is the consumption. I'd have to go confirm at the MDH website.
So that
I'm pretty sure it's based on a 6-ounce fillet.
a 6-ounce
That's the
fillet
dose.
for a 50-year-old who's probably not going to have.
Right, so you'd be in the low risk category, someone who's not anticipating having a child.
And
That'd be a big surprise.
everyone would be impressed.
It'd be a lot worse than mercury.
Yeah, but it would be once a month, 6 ounces once a month, or
Yeah,
once
once a
every
week.
It's probably
once a
between the once a
week.
week and once a
Once
month.
a week
I'd have to
or
go
once-
look at the numbers
Yeah, you'd have to look
precisely.
it up, but you could find that pretty easily.
Yeah, I'm sure
But
it's
when
in
we
that
think
range.
about that for someone like me, and I know when we go camping and we catch a bunch of fish and we eat a bunch of fish, we got a pile of fillets. It's eaten before they're, nobody there, the kids, everybody's eating that fish.
Now, I'm here because this is a huge problem. This is a huge, and it's a global problem.
Mhm.
We have substantially reduced access to a sustainable food supply.
Right, that to me is the
Vis-a-vis
most important
the vast
thing.
manipulation of the mercury cycle.
Can you say it again? We have a substantially reduced
Access to a local sustainable food supply.
local, sustainable, no, because that's the core of it.
It is absolutely cool.
That's exactly what we're telling people, is you can't eat the
You
food
cannot eat the
that
fish
the
in your
earth
backyard.
has
That's what we're
put,
telling people.
yes,
And we're saying, sure as heck, don't feed it to you if you're pregnant. Yeah,
When
it's
you
great-
need
It's got great
protein.
omega-3s, but gosh, don't eat it.
So I've got a question, and either one of you could answer this, I'm sure both of you know the answer to it, but I just want to make sure that I understand this. My understanding is that when it comes to wild rice, it's hydrogen sulfide that actually causes the toxicity?
It appears to be.
It appears
Yeah,
to be.
that's John Paster's work.
And so, but it's different for mercury methylation. It's actually the process of reduction that causes the methylate, it's the reduction of sulfate to sulfide that some part of that, something in that process creates methylmercury.
That's right, that's this process
So
right here, where
it's
the
not-
bacteria are intermediating this.
Right. So
They're
it
basically
seems that
replacing
with,
the sulfur group with the methyl group.
so it seems that with wild rice, it's a good thing for change back to the sulfate from sulfide, because the sulfide itself is toxic. But for mercury methylation, the change back to sulfate is not good, because it makes it available for the reduction again, and it's the process of reduction
Well,
that causes the problem.
Not quite. So in all cases, it starts out as sulfate in the water, because the water has
I
oxygen.
understand that.
So what happens in these sediments, in these reducing sediments, it gets converted to sulfide when
Right.
in lack of oxygen
Right.
from bacterial activity. That's the form that becomes toxic.
Right.
It's
and
that
but
same
that's also
environment
that
that's
same-
promoting methylation, actually.
It's the environment, but it isn't the reduction process that creates the methylation.
It's the same
Or they
bacteria.
don't know.
It's the same bacteria.
I tried to find the answer to this online, and what I kept finding was, we don't know yet.
I've tried to find this too. I couldn't remember it to say it, but
Would you say, Joel, it would be correct to say that it's the same sulfate, it's the same conditions, anaerobic, so forth, and it's the same bacteria, and one of the outcomes, one of the waste products is sulfide, hydrogen sulfide, for example, that damages the wild rice, and one of the byproducts is methylmercury that damages
Yeah,
us.
right, I mean, I've got all that, I understand all that. But then I got to the point where you were saying, when you were talking about the fluctuating water levels in the reservoir,
Mhm.
that one of the things that happens then is
Sulfide
is oxidized
becomes,
again to sulfate.
that's right. So when you take a wet sediment and you dry it out, you get oxygen.
And so that, and then that leads to the situation where you have more sulfate available again for reduction.
That's right.
But it seems that with the wild rice,
But that sulfate would be, the reason why it's available, so then what happens is that sediment gets wet. It becomes a reducing environment again.
and it's going to switch right back to
Yeah.
sulfide again.
Yes.
Okay, all right,
Yeah.
okay, thanks.
so when it dries, it becomes sulfate, and
Yep.
then when it gets wet again and gets flushed out, it's sulfide again.
As it becomes wet, it sets up the environment that allows for microbes to start reducing.
Methylating it.
Right. And one of
Is
the first things
reducing
that happens is,
methylating?
see, you have to remember that there's actually, biogeochemically, there's a series of processes here So the first thing, the most energetically advantageous thing for bacteria to do is to actually consume the oxygen. So consuming sulfate is actually way down on the list of down on the list of energetically things that bacteria want to do. It's more advantageous. Now these are different whole communities, so don't think of it as the same bacteria. But so if you go into the sediments, the first thing you lose is the oxygen first. So it's always a transgression. So before there's always a transition. First to the oxygen gets consumed, and that's when after that then we start seeing sulfate converted to sulfide. So the very nature of setting up this aqueous sediment starts its transition to being a low oxygen environment. That's the first step.
And then I also was curious about the iron reducing sulfate. Do you know anything
I
about that?
completely deliberately avoided that.
That's really new stuff. I don't know that anybody. That's
Well,
really new.
that paper, the paper I have is from 2006. That's quite a while.
2006,
Oh,
but it's
no.
really just now getting a lot of
And it
I
hasn't
sat
been
through
well
a
replicated.
presentation on that, and the thing that worries me is that it seems like an easy answer, along with the thing I'm really worried about is they're going to start talking about, well, then we need to dry the wetlands out entirely and get rid of them if they're really, really, really obnoxious.
I mean, right, so there's an extreme view here which runs against all the other management policies we have in place,
Uh-huh.
which is opposed to, yes, essentially fill wetlands and reduce methylation. I mean, I suppose that's true.
But a lot of those wetlands are methyl mercury sinks. Depends
Yes.
on transport.
It
Transport
does.
becomes so critical.
I'm
And
thinking
we have
of this
already lots of
concept.
other environmental policies to protect wetlands. I don't think anyone's really.
I'm
considers
thinking
this.
of how Tom Bach and McNamara might interpret this, and what they might do with this kind of logic.
He's working on the science, not the politics.
Yeah, I can't speak to
Yeah,
how
we're-
the politician
that's,
interprets this.
No,
So-
But I think it's,
but you know what I'm
I
getting
think at
at.
least from a scientific standpoint, whether or not actually trying to reduce the influence of wetlands on the system is a net good thing is, I mean, everything that we know about how wetlands function would suggest environmentally that would push the system in the wrong direction.
Yeah.
When you talked about currently some methyl mercury can be transported just in the water column, is that attached to organic particles? Is that how that gets transported?
Mostly, but remember, it can disassociate. So it can be attached as a particle, it can also be free, and it can be colloidal.
It can, okay. So all three forms can be floating in the water.
That's right. And of course, I mean, here we're talking about chelation processes, ligand binding on compounds, which is some of the toughest chemistry, interacting with organics and metals. So yeah, but it's out there. And I don't know if you remember from Gary's talk at the Team Dale related gathering we had this past fall. One of the things he pointed out was that there is actually an equilibrium dynamic going on here. So at some level, one need not even invoke bacteria. Some of this just happens at an equilibrium basis. It's at a low level relative to bacteria-driven methylation. But we don't need bacteria to even have methylation. It's in rainwater.
If we could stop the mercury pollution tomorrow, how long would it take for the system to recover?
It's a lot of factors. It comes down to ecosystem type,
All those things
the
you've been talking
residence
about.
time, how much of this legacy material has been stored in the system. They got a lot of press. This paper came out this fall, some study on some Voyagers lakes where they found out that because of reductions in long-term atmospheric input, some lakes have responded and some simply haven't.
Yeah.
So, big picture trends, broadly speaking, mercury's been going down. You look at the Great Lakes, in the big picture, from the 60s to now, mercury in the monitored fish in the open waters of the Great Lakes has been trending down.
Oh, really?
It's been trending down faster on our side than on the eastern end of the Great Lakes, which most people agree reflects regional deposition patterns. Erie, Ontario get more mercury to surface waters than on both an aerial basis and on a total input basis in Lake Superior and Huron, Michigan. Now, there's been some trend reversals recently that have made some media news. In some of these lakes, relative to the 60s, they're still way down here. But there's been some evidence in some of these lakes there's been a recent uptick over the last 10 years. It's small, but it's over many years detectable. And there's a lot of hand-wringing about the cause of that.
And some of those lakes are in the, like, inland from Silver Bay and in that area.
Yeah. Yep. Some of them are inland, some of them are offshore. And this gets into some of the hard aspects of the biota pieces too. Question then arises, is that reflecting an input, a mercury bioavailability process, a bacterial process, or there's something different about the fish? And so you see that uptick and you don't know which one it is yet. And in many cases, the way the monitoring programs are designed, we can't simultaneously collect data on all those things. So we have to run the additional studies to try to understand what could be driving it. Otherwise, all we have are a bunch of hypotheses.
When you think about the mining that's planned in the Duluth complex, do you think it will have, if several mines are established in that sulfide-bearing land, do you think it will have an impact on Lake Superior if it's in the Lake Superior watershed?
I don't know that we know that yet. The studies, a lot of the work that's going on TMDL is designed to help us understand exactly that.
Mm-hmm.
A well-done TMDL study should hopefully help us understand what kind of linkage there could be between some change in mining behavior and inputs in the region of the lake. I would say a real challenge there in understanding that beyond just the science and having a good conceptual theoretical model that deals with all these things that can influence mercury, is that one of the things we don't have are good long-term data sets in the western arm of Lake Superior for mercury and biota. So in some of these cases, you want a system that's responsive to a before and after state, and we should be all mindful of whether or not there's a before data set that would allow you to say something about a future, whether the future world we created changed it.
So you're saying that if you know that you're in the process of, say, copper nickel mining, if you're creating 95% of the material you take out of the ground is waste, and 1% or 5% or whatever it is useful metal, so you're stuck with all this waste, either piled on the ground or submerged underwater or supposedly treated with reverse osmosis. You're saying that it's difficult to judge whether that would be a problem or not, or
She's talking about for Lake Superior.
well, Lake Superior, obviously, that's where it's going down the St. Louis River.
Yeah, Lake Superior's a big lake.
I'm interested in both,
There's a lot of
but
water in that lake.
Yeah. Yeah. So this is what the TMDL is supposed to answer. So the TMDL is really about is sources and fates.
No, my question was...
So the question would be, in that context, you're talking about changing behavior, a new mine or a new way to mine, or a new way to handle the waste, like this idea about reverse osmosis. What you'd want to know is straightforwardly, what's our best estimate about what that new contribution would be. And it wouldn't just be mercury, as Lance pointed out, it would be changes in other chemistry as well, including obviously sulfate.
And land use.
And land use, and in wetlands, and flow. These things are also huge hydrologic impacts. They're going to alter the routing of whole wetlands. So you have to take all of that into account too, and understand how that fits in. And these ecosystem models are not established for the basin. There's nothing you can just pick up and say, if we take their mining numbers and threw them over here, turn a crank on a bunch of numbers, bam, here's what happens in St. Louis River. That does not exist in the world.
You see what we have looked at this, today, and you start to get an appreciation for the incredible complexity of the real world. And then we come up against a couple of things, one of them being a St. Louis River TMDL. And it's very easy to say, we don't have enough information to really do a St. Louis River TMDL, and that's what the MPCA said, you know, a year and a half ago. But on the other hand, we've got mine permits that are happening, not just polymet that may happen, but we have mine permits that are happening in which we have to make management decisions on how much of this we can put together in the best possible way. And there is no one solve-it-all model. The warmth model attempted to, but the state was quite critical of that. So we are going to make those management decisions, no question about it. We did last year and the year before. We continue to make them.
Yeah, it's like, just because we don't have complete information doesn't mean that we're not, that we shouldn't make decisions based on whatever information we've got so far. You
And
know,
I
it's
would
like,
say-
it's no reason to not look at what information you do have just because that information isn't complete.
I would say
You have to
we have
use
a
the
couple of
information you
people
got.
that, I'm sure, Joel, you know, that are in a good position in the Pollution Control Agency to make those management decisions and make them pretty well. And that's Bruce Munson and Ed Swain. I mean, they're very, very good. However, they're not the ones that write the permit. They have input.
Yeah, I don't know,
There's a
I've been
question.
reading the old Minnesota Environmental Rights Act cases, and, you know, there's some really bad law out there. There's one case where the PCA said no to a permit because it would release too much, it was about dioxin, you know. And even though they didn't have a standard for that dioxin, they said no to the permit, and the court overturned them and said that if a polluter is going to meet all the standards, they have to issue the permit. And I'm wondering if that wasn't the point at which things shifted at the PCA, because I've seen a shift there, you know, to the attitude of, you know, we're permitting everything. And I don't think that that's always been the case.
You know, in some sense, I think our best tool in our battle to reduce the mercury in our fish has been wild rice. We've got a standard there,
Yeah,
and
and it's
not,
already in place,
you know,
and so.
it's not perfect for methylation, but it's sure a lot better than no standard.
When they talked about that today, Carly Moline got up and said, we all know that's a ruse. Nobody bothered enforcing it. They just are trying to stop this mine. And she said, they don't care about this with relationship to wild rice. It's all made up. She said that.
Well, people say a lot of things.
So we now just are done with the science. Now it's all politics. We're done.
No, the translation of
I
this
wanted- I'd
to
like
those
to-
people is what I'm getting at, is how do we help them really understand?
Some of those people, there's no point, Kristen. They don't want to understand.
We have to make them understand.
No, they're not gonna, they are never, ever, ever. You have to accept that there are people who do not want to understand, and there is nothing that you will ever say that is going to make them want to understand.
Well, then the political process is for nothing.
No, it's not, because there are other people out there, there are other politicians who you can help understand, but there's a certain subset that have already made up their mind, we're not gonna change them, you know. I mean, that's not hopefully not the majority, you know, but I mean, where it sits right now, it's pretty bad. You know,
It's Lincoln's thing, you can fool some of the people all the time and all the people some of the time, but you can't fool them all of the time. Sooner or later, the truth comes out.
well, they want to be, I mean, they, you know, they're going on what they want to believe. People believe what they want to believe.
Right.
And,
I know what it is. The majority of the citizens have to understand this stuff. We have to make
And
it
it is
less-
so complex, we kind of
We have to
won
make
that
it less
acid rain battle.
complex.
We won that because the geochemistry of that was so much simpler.
Yeah.
But now, just think about the things we've gone over today. And think about where your average voter is in terms of their scientific level of understanding. There is a tremendous amount of educating that has to go on, and it is not going to be easy. But they're not interested in paying that price.
You can't educate the people, you can't go up and tell a bunch of young women that may be conceiving that if they drink this water, the child they have may be developmentally disabled and cost them $100,000 a year to take care of them. You just can't go, and as far as I'm concerned, that's what the big public health message has been, that you cannot discuss. Right or wrong, that's my opinion.
Not drinking the water, but eating the fish.
Well, eating the fish.
They're certainly working on getting that
Well,
message
there's
across.
things-
Is methyl mercury ever suspended in the water so that it could be in the water you drink?
It's the bioaccumulation factor that makes it dangerous.
Yeah, I don't-
It's
That's
only
your
eaten.
Well, they
It's
measure
only
the
when you
alcohol
eat
in
animals.
the
But
Oh,
can
yeah,
you-
well, we'll come back to this. Our first order,
Uh-huh.
this is the problem.
The water is at a really low level.
Even when the water is at
mean,
incredibly low levels,
there
parts per trillion, it ends up in the parts per million, 6 orders of magnitude higher.
So I'm
in
never
altitude.
gonna get a problem with drinking the water.
There's
But,
other toxins
Not unless you
in
go
it.
to a
Well,
lake.
Other
that's
toxins,
on-
but out of
that's
methyl
fine
mercury.
on it.
I'm sure you could find some water bodies.
Yeah, California, the
world that-
mercury mines in California
Yeah.
have really high.
Or
Levels in the
perhaps some
water.
shallow
But
lakes
our,
in
our waters
Minnesota.
are, your tap waters, et cetera,
But like some shallow lakes in Minnesota.
I don't think so.
Not
I've never
possible?
seen numbers like
Okay.
that from this public.
Okay, so that
No,
we're
that's
we're
what-
only getting it from what we eat, which is what I understood till I
The
got
vast
interested
majority
in
of
what
exposure.
Len said. I thought, oh, great. But it somehow we have to make this clear to people how small that amount of fish is.
Okay,
And
so
the
this
impact
is,
it can
this,
have.
that's a discussion for another time and
Yeah.
place. And we were.
I think we're over.
We didn't get to, this,
A lot of
this,
things.
yeah, right. And so I'm wondering, what I would like to do is, get a bibliography list. It'd be great to get one from,
That'd be one.
okay, as well as from Len, from Joel, as well as from Len. And I'll put it out to people, and then let's figure out a way to make those available to people. Maybe we'll just have them, a collection someplace for a certain amount of time that people can go in and read them, maybe at the MEP office or someplace.
I've been able to download a lot of them while we were
Okay,
sitting
that's
here.
great. Okay.
Through the University of Minnesota. But
Okay.
the other thing is, I ordered the book while we were sitting here, so we
Okay.
can share that too
Okay.
amongst ourselves.
But the
up
thing
here.
is in, how to make it, have it available so that everybody can look at it if they want to. And then I'm wondering if we want to
Good job.
plan another time to get together with Len to talk to him about questions that we have. Is that something you might be willing to do? No PowerPoint, no, it's just a few people sitting around a table and talking.
Joel, would you want to come to something like that?
Yeah, let me
Okay.
think about it.
In the context, but I could certainly, my role here, I'm always in a weird position in these sort of things.
Yeah.
My role, I'm happy to come in as a scientist.
Sure.
I mean, you as a group of
Advocates.
advocates can, what to do is up to you. But how science fits in, science isn't.
Might
the way
turn it
we-
off.
yeah,